Mario Capecchi from the University of Utah. We're very honored to have him here, and Ron will introduce him further.
My other friend from the University of Utah is Mario Capecchi, and I love talking to him. Always learn some new stuff, and I think you should be very interested in what he's going to have to say. This, or do you want to talk?
Do you want to use this one? Or? This is good? You can hear him in the back? Yes? Good. First of all, I want to apologize because I don't work on CFS. And I'll tell you a story about a disease that has some similarities. And the similarities that you want to keep in mind are, one is that inflammation is going to be a big player. Another is that anxiety is going to be a big player. And the third is that immunology is a big player. So those are the take-home lessons. You may leave now. No, so we got into this work by serendipity. We work on Hox genes. And Hox genes are an interesting set of genes because they're involved in making the body plans of all multicellular animals. And in particular, what they're interested in doing is giving you a sense of direction, your axes, what we call an anterior-posterior, which is your main body axis, and also minor axes, for example, your limb, proximal, distal, and then up and down on the hand, okay? So all of these axes are encoding by these molecules, by these proteins, which are all transcription factors. That is, they turn on and turn off genes. And the nice thing about them is that what you're looking at is 4 sets of genes. That is, and on the very top is what's actually in Drosophila. In Drosophila, there's only a single set, and then there's been a quadruplication of that entire complex, and then addition of more members at the far end. But what's interesting about the genes is that where they are on the chromosome tells you exactly where they're operative in your body. So genes at this end of the picture operate at the anterior aspect of your body, the genes all the way at the opposite end, at the posterior end of your body, and then everything in between. And they're responsible for making the analog, the beginnings of all the different cells that are going to make all the different parts of your organs. And then you make them in the right place, and then you hook them all together, and now you have a working human being. And this is particularly a complex in mice, but it turns out it's exactly the same in humans. So the other thing, my patients are usually mice. And the one thing that I also want to get across is that with respect to genes, mice are 99.9% the same as you. Okay, so whatever we're learning in the mouse is likely to be also applicable in humans. So the first thing we did was simply distribute these genes among my graduate students and say, you're responsible for this 3, and the next one's responsible for the next 3, and so on. And Joy Greer was responsible, one of her projects was responsible for HOXB8. So where is that? Whoop. Can you see, whoops, no, come back. I don't know if you can see a light there or not. Oh, yeah. HOXB8, that's the gene right there that she's responsible for. She knocked it out. We developed a technology to be able to specifically knock out that gene and then ask what happens. And that tells us something about what that gene is doing. In the absence of the function, what's happened to the body, and then you can tell what that gene's up to. Okay? She knocked out that gene. She looked at the mouse and it looked perfectly normal. It was running around, and that was really quite surprising because most of these other genes, even if fortunately you get 2 copies of every gene, one from your father and one from your mother. You knock out one, usually the other copy is still okay. But in these cases, often if we knock out one, we still see something. And here we knocked out both copies, and we didn't see anything with respect to the body. And that was quite surprising. Okay, so she's a clever woman. And so she said, well, if I can't see anything physically, maybe there's something weird about the behavior. So mice are mostly active at night. And so she gets out her infrared camera, sits there 24 hours and takes movies. And then looked at everything as what this mouse is doing. Running around the cage, upside down in the cage, drinking, walking around, running, all of these different activities. She's taking movies and then simply looks frame by frame and jots down how much time is it spending doing this and that over 24 hours. Okay? And what she found was everything was the same except one behavior, and that was grooming. They spent twice as much time grooming as normal. The other interesting fact is that there's a sex bias. Okay? Women mice spend a lot more time grooming than men mice. So that's an interesting fact. And then the other aspect is that all animals groom. And how we groom is actually quite related. The syntax, what we call the syntax, is the same. You go into the shower, lather your hands, and start working on your head. And you work down your body. And if there's anybody that doesn't have that pattern, please see me afterwards. But these mice spend a lot of time grooming, and it becomes pathological. And so they continue to groom until they actually start removing the body hair. I don't know if that's shown here. Here's a little movie. Let's see, we better activate it. Oh, come on, okay. And what you'll see is it keeps on grooming and grooming, and when it moves to the side, you'll see that it's actually removed the body hair. There, okay? And then it continues to groom until it actually has lacerations, and at that point we put it to sleep. Okay, because, and this is very similar to patients with OCD, and they, for example, wash their hands and continue to wash their hands until they have lacerations. And it's also very similar to what's called an OCD spectrum disorder called trichotillomania, where these children actually compulsively remove their body hair. These are both very common. 3% of all populations that have been looked at, many populations have been looked at, have this behavior or behavioral pathology. Okay. So we looked for our gene, where is it functioning in the brain? And we looked very carefully and we couldn't find it. So we have a trick. We can take a gene from a jellyfish, not a snail, but a jellyfish this time, that actually makes a protein that fluoresces green. And they make proteins that fluoresce red and they're blue, and all sorts of colors. And you can take that gene and put it in the middle of another gene, and then wherever that gene functions, then those cells become green or red or yellow or whatever you want. Total spectrum of the universe. Okay? And so that then, and those cells will then stay that color for the rest of their lives. It's what we call a cell lineage marker. And that tells us now where is that cell in the brain. And that gave us an enormous surprise, and that's shown in this slide. And so here we're looking for green cells, and here they are in the brain, and we look at them, and they're not neurons. Everybody, what we were expecting is neurons, and these are actually microglia. And here's a marker that labels all microglia, red, and then what you can see, these are yellow, which are both red and green, okay? Good combination of those two. And this is a microglia marker, but what you can see is not all microglia are green and red, okay? Only about 30%. So that, to us, tells us there's two populations of microglia. Okay. So what I'm trying to tell you is Microglia are immune cells. They're blood cells, okay? For example, in adults, they're made in the bone marrow, and then they go travel all over your face, all over your body, and then do their thing. But in this case, what we're saying is that microglia, an immune cell that goes to the brain, is responsible for behavior. And then when it has a mutation, now you have altered behavior. Okay? Try to convince somebody of that. And we've had a difficult time convincing anybody of that. But I think it's true. So that's a new rule is that defective microglia are somehow affecting neural circuits, modulating those neural circuits, so you have a behavioral output that's not normal. So, but it made a very strong prediction. You can do a bone marrow transplant because these are blood cells. We can radiate the mouse and then kill its own immune system and now supply a new bone marrow cells, and they will colonize and then produce cells. Okay. So we're going, whoop, uh-oh, what did I do? I did what my colleague did. Uh-oh. Okay. So, first of all, we just wanted to see whether green cells get into the brain. So we use green cells, put them into a radiated mouse and look into the brain, and they do get into the brain. That says, okay, that's good. And then we did the actual experiment. So here's a mutant mouse 3 weeks after bone marrow transplant. Bone marrow transplants don't take place right away. It takes a while to generate enough cells to do something. And what you can see, it's removed its body hair. And if I showed you this a week later, you see lots of lacerations. But 12 weeks later, now all of the lacerations are healed, all the hair has grown back, and now this mouse no longer has that behavior. So it's been completely reversed by a bone marrow transplant. Okay? So again, indicating that these cells are likely to be causative for that behavior. And we can do the opposite, that is, we can put mutant bone marrow into a normal mouse and give it the pathology. Okay. So this, to us, suggests that indeed microglia immune cells are responsible for the behavioral output. And we're continuing that to be able to try to prove that. The other thing we've looked at, because it was quite controversial, is where do these cells come from? And what we found was that they come from a different source. Normally, where these cells come from is in the yolk sac, very early in embryogenesis, and go to the brain. What we found, these particular cells don't come from the yolk sac, but then start in the yolk sac, then go to the fetal liver, and then go to the AGM, and then finally get to the brain many, many days later. Okay, we're going quickly. Okay. So here we're simply looking at where do these cells come from and when do they arise, and what we can see is that they actually are made in the yolk sac, go to the AGM, and then get amplified, and then go to the fetal liver and get more amplified, and then finally go into the brain and do their thing. And this simply collaborates that. We can ask how different are they from the other cells that people know about, and we find that they're quite similar. There are about 21 genes different in those, and that's compared to thousands of genes, that's a small number. And then finally, we asked how do these cells behave? Are they different from the other microglial population? And the answer is yes, and I'll show you just this little movie. What we've done is to use a laser to destroy cells right in the middle. And then what you can see is yellow cells and green cells send their pseudopodia to the mutants, to the defective site to try to correct that damage. And what you can see is that the green ones and the red and the yellow ones get there at exactly the same time. So by this assay, they're very, very similar. In another assay that's common, another thing that microglia do is often when you set up your nervous system, you have many, many more connections than normal. That is, you make twice as many connections, and then the strong ones are kept, and then the weak ones are removed. And who are responsible for doing that? Microglia. They can actually tell the activity, what's going on at a synapse, and say, this cell is not very active as when it should be, and the system goes to those cells and removes them by eating them, and then lets the other ones remain and become strong. And that's shown here. And the other thing that's interesting is that the two populations aren't just randomly exposed into the brain, but they actually know where to go. And in particular, what we're looking at is the ratio of our cells, which we call HOXB8 microglia, because they had expressed HOXB8, and the other one non-HOXB8 because they don't. And what we see is that the ratio of those cells varies in the brain, the location of the brain, and where do they reside? They reside in that circuit, neural circuit, that is responsible for this behavior. So that is, they're in the right place to be able to control this circuit. And now we're trying to figure out how do microglia actually talk to neurons and modulate their behavior. Thank you.
Thank you very much, Mario. We'd like to welcome up our next speaker. There he is. Mark Davis from just down the street at Stanford, who will be answering a very compelling question of whether, or answering or asking, well, maybe a little bit of both, as to whether ME CFS is an autoimmune disease.
I've known Mark a long time, done a number of projects with him. We've even set up a company together. A lot of people ask me, are you related to Mark? And I said, of course, we're all related. People tend to forget that. So I consider Mark one of the best immunologists in the world. And you can tell that by visiting his office. It's stacked with papers. And you have to worry about having a landslide and getting buried. And so I'm really, really happy that he's entered into this field, and I think what you're going to hear is absolutely phenomenal. So pay attention.
Thanks, Ron. Yeah, we're Davises are everywhere. That's so as the only card-carrying immunologist in the show today, I thought I should take you through a few things, and partly it's going to be important understanding what we've done and the clues that we've developed in terms of this thesis of is this an autoimmune disease? Why do we think that? What's the evidence? And for that, you really just I just want to take you briefly through the immune system because it is complicated. Some people black out when they start hearing about the immune system and its complications, but it's getting simpler all the time, which is that means we're understanding it and we can reduce it to just a few statements like there are a bunch of white blood cells that do different things, and they have names and functions, and they talk to each other with cytokines. So key ingredient in the immune system, different white blood cells talking to each other with different cytokines, coordinating activities. But at the end of the day, it's the individual cells and whatever it is they're supposed to do that make things happen. We generally think of the immune system as two components, although they interact as well. There's the innate immune system, which is sort of the fast-acting, oh my God, something's happened. I'm bleeding. Let's have some inflammation so we can get more white blood cells into the inflamed part, the injured part. And it can be injured through with just a wound or more often an infectious disease is coming in. And then a little while later, you can get this adaptive immune system, to kick in, which are the lymphocytes. And lymphocytes are making these very, very specific receptors that essentially are the body's solution to our intense evolutionary problem with respect to viruses and bacteria and pathogens, which is that they can replicate sometimes every 20 minutes. We replicate only every 20 years. So there's no way we can win an evolutionary race, you know, if we start at the same point. So we cheat, and we cheat by making every possible receptor against things that we've never even seen before, and maybe that have just evolved on Mars a week ago, and then they come in and there you are. But you already have antibodies and T cells specific for those. So how does that happen? That's one of the remarkable things about the immune system is that it creates diversity at the gene level. It's the genes for antibodies and T cell receptors are split up into pieces, and where there are many alternative components. It's like an old Chinese restaurant menu. You can pick one from column A, and one from column B, one from column C. At the genetic level, and then these come together in the chromosome, in the DNA, fuse together, very specific mechanisms. And at the end of the day, then you have billions of possible sequences just in those bits of the molecules, the big proteins that are responsible, or the business end, are binding to whatever it is they might want to bind, which is sort of every possible thing. And some of the numbers are pretty amazing. As I say, when you put all these things together, both T cells and B cells, you get very large number, billions and trillions of possible combinations, just because of the way it's put together. And this all happens at the DNA level, and once it happens, it's irreversible, it stops. In fact, the whole machinery for rearrangement gets activated during certain times in a response or in development, and then once it happens, it stops. And once you have a good antibody gene or a good T cell receptor gene, the whole recombination process stops, and then that is what that cell has. But the cell next door has another one, and the one in the whole crowd will be expressing billions of possible variations. Now, let me throw another complication at you, which is that's the story for antibodies, and it's also the story for T cell receptor genes. But T cells are a little trickier than antibodies in the sense that they're actually recognizing peptides. You just heard about poisonous peptides. These are just regular peptides that have been degraded out of whole proteins. But it's also the beauty of this system is that every protein gets degraded, whether it's a protein that belongs in your cell or whether it comes from a pathogen or a virus. So there's not any good way in which the pathogen or virus could disguise the shape of its peptide, or it can disguise the shape of its proteins, but once the protein gets degraded into peptides, it no longer has the ability to fool the immune system's detection system. And so that's where T cells come in, because they are recognizing little peptides that are stuck in this big molecule that is often been described as a moose head. And the T cell receptor on the T cell is on, it's coming down and contacting this combination of little peptide and this moose-like MHC structure. And then between cells, which I can barely see, but you can see a representation of a T cell receptor recognizing a particular peptide MHC. There are other complications, including that we have ways in which we can look at peptides that are made within a cell and peptides that are made that are coming from proteins outside the cell. But let's, I think, move on to the other key thing about basic immunology that I want you to realize is that how does this whole thing work? If you're making billions of possible specificities, how does that actually do anything? Don't you get lost in all the complexity of that, or don't the cells get in each other's way? No, because what happens is that a whole collection of T cells is expressing these billions of possible T cell receptors, but only one that has a receptor that can recognize, in this case, a specific virus, is stimulated enough to trigger cell division. And that cell division then makes a whole bunch of daughter cells, represented there, that all have the same DNA rearrangement for that receptor, and therefore they all have the same specificity. So this is the amplifier mechanism. This was a big deal, discovered 60-some years ago in immunology, and it's a way you pull out the T cells or B cells from the crowd that have just the specificity that you need for whatever the pathogen is or whatever the stimulus is. Now, as Dr. Light said earlier, the system makes mistakes. And sometimes if you make a response against, say, a virus, there can be a cross-reactivity to self-proteins. And that can sometimes trigger autoimmunity, that you start off making a perfectly reasonable response against a particular virus, but if things go wrong and there's a similarity between that viral peptide or protein and something in your heart muscle, for example, you can have a serious autoimmunity to your heart. You can have serious autoimmunity to your joints, to every, various tissues. Juvenile diabetes is autoimmunity to your insulin-producing cells in the pancreas. So there are lots of problems where things have gone wrong. So let me take you through the chronic fatigue data, and I'll be referring back to some of these things. One just is just recently published in PNAS, Proceedings of the National Academy of Sciences, largely the work of Jose Montoya. I helped, but it's really his study. He looked at almost 600 people, 200 chronic fatigue patients, and almost 400 controls, age and sex matched, and for a lot of immune biomarkers, particularly cytokines in this study. He's also looking at other biomarkers, but this is the sort of first tranche of this a very large study. And I could say that that this study was funded solely through philanthropy. And that's our big problem, as Ron has mentioned, in terms of getting the attention. We finally did get the attention of NIH, and they are finally putting more serious money than they have before into this sort of thing. But it takes time, and for areas that are not well understood, there's a tendency to dismiss them, to ignore, hope it goes away, not going away, as I'm sure you know. But one thing philanthropy can do that NIH has trouble doing, NIH relies on peer review. And peer review means that they assemble a jury of people that have apparently the same skills and knowledges as the person applying for the grant. Which works pretty well in areas that are well understood and there's a clear path forward. In areas that are not well understood and there's no clear path forward and typically massive confusion, just imagine Elon Musk going and applying for a grant from Detroit 10 years ago to start Tesla. That's kind of somewhat analogous to where we are now, is that there's confusion, and then there's no consensus, and so forth and so on. So anyway, long story short is that in this study, we saw remarkably of 51 cytokines that we can follow in the serum of chronic fatigue patients, 17 were elevated, and they're largely weighted towards inflammatory cytokines. And they also correlated, as I hope you can see here, we're looking at mild, medium, and severe patients, ill patients. And what you can see in every case, there's a progression from mild, having lower levels, to medium, higher, and then severe, even higher than that. So this really says that there's a lot of inflammation going on. And inflammation takes various forms and means various things, some of which I think we're still understanding. But this is definitely an indicator of inflammation, of some kind of systemic inflammation, and that this is typically coming from the innate immune system. But we're interested, we have done a lot of work on T cells, And we're particularly interested in whether there's a T or B cell role in this, and something we could trace back, which you can't do with cytokines, but you can trace back with B cells and T cells because the receptors are so specific. And this depends partly on a novel assay that we developed at Stanford a couple years ago, where we can sequence at a single cell level, we can get T cell receptor sequences from any patient, any tissue, any disease you care to look at. And one of the things that came up very quickly in this, looking first just at some T cells in cancer. So in many cancers, T cells infiltrate and proliferate within the tumor, and they often are specific for antigens in those tumors. So one thing we've seen here very clearly in this colon carcinoma, there are a lot of T cells, and by sequence analysis, we can tell that, for instance, there's 60 different T cells from this tumor that all have the same T cell receptor in red here, and then about 40 that all have this blue one, and so forth. But what you can see is that more than half of the T cells in this tumor have expanded clonally. That is, there's something there that triggered cell division, and that's why you're getting the same T cell receptor. Whereas in adjacent tissue from the same patient, there's almost no clonal expansion. So something's happening within the tumor. We've also seen this in a mouse model of MS, where kind of a classic model where as you induce the disease, you see clonal expansions of T cells with the different days, and both in the blood and in the central nervous system. And that's also CD8 cells. We also looked at MS patients in a collaboration with a group at UCSF. And here also in control healthy subjects, we see almost no CD8 clonal expansion, but in almost all of the MS patients, we see massive T cell clonal expansion in the blood. So we can look in the blood, we can see evidence of really strong T cell proliferation and division, which we associate with specific responses. Similarly, in Lyme disease, we have another study in Lyme disease. We see the same thing, especially in CD8 cells. That's not CD8 cells. Those are CD8 cells. So you just see a lot of clonal expansion. So this was one of our first things to look at in terms of chronic fatigue, and particularly we started looking at it because in work with Jose Montoya, we found that we look at T cell receptor sequences from CFS patients, we see a lot of similar T cell receptor sequences shown by these dots here, and not random like these similarities here in controls and so forth. So that indicated there are some specific T cells there. And so we dug deeper. We looked at 6 patients which had particular HLA types, and we put our methodology to work. We ended up sequencing about 1,000 individual T cells. And sure enough, the CD8 cells from all of these patients showed evidence of a marked clonal expansion, that something's going on that's stirring these cells up. And particularly there's an antigen, almost certainly an antigen-triggered event, that is causing them to divide. There could be multiple antigens, but there certainly are some more than normal. So in summary of all this, I showed you, not here cancer, but in cancer, in infectious diseases, in autoimmunity, and in chronic fatigue, characteristic is CD8 clonal T cell expansion. And at least in some cases, we know those are specific for the disease. So we also have now gone on with these T cell receptors to look for the antigen. And here we're collaborating with Marvin G. and Chris Garcia because they have a nice yeast display system to find the antigens. And so far we have just one hit out of the first couple T cell receptors, and we're working that up to try to understand what's the antigen, and does that mean, does that indicate some particular antigen, some particular pathogen, some particular tissue, or both, which is sort of what we would expect. If it's an autoimmune disease, you'd expect to see that this originally was against some pathogen peptide, but that it cross-reacts with some self-peptide. maybe in the microglia, maybe, maybe somewhere else. So anyway, this is where we are right now. We see evidence of really major activity in terms of inflammatory cytokines in the blood, very reproducibly. Of chronic fatigue patients, there's a correlation with disease severity, so we know something's stirring up the innate immune system. It could be a pathogen, it could be some sort of auto-inflammatory thing, which is not that well understood. But what it also could be is part of a whole immune response that involves T cells as well as the innate immune system. And T cells make a lot of cytokines, and they could make some of those cytokines. But particularly what I think is most exciting about this sort of line of approach is that if we know more about the specificity of these CD8 T cells that we're seeing expanding, it could lead us directly to a pathogen that might be the precipitator of the disease, and also autoimmune peptides that could tell us more about the kinds of pathological effects that people are seeing in this disease. So that's our hope and our mission, and we have many people to thank, particularly though most of this work is done by Santosh Kumar and Elsa Sola, postdocs in the lab, with help from many others, including Connor Gajan, and lots of great collaborators, including Ron and Jose Montoya and others. So thank you.
Thanks very much, Mark. We are now on coffee break. We will resume at 3 o'clock for the home stretch.